Evolutionary Ecology 1. It's the first of many evolutionary ecology sessions, and we have an exciting list. It's time to start. We have an exciting list of talks. Okay, we're starting. I'm Emily Behrman, and today I'll be talking about how strong seasonal selection results in rapid life history changes in Drosophila. There we go. So variation in spatial and temporal gradients maintains genetic diversity in nature. Environmental gradients, particularly along latitudinal lines, are commonly used to evaluate the associations between genotype, phenotype, and the environment that are driven by natural selection and thus reflect the adaptive process. The mechanistic basis that underlies latitudinal declines, however, are unknown for the most part. But the general hypothesis is that latitudinal declines reflect a fitness trade-off between genotype and the phenotype and the climatic environment. In many respects, seasonal time and latitudinal origin reflect parallel gradients in terms of environmental parameters. Therefore, you would expect collections from early in the season to reflect those of northern populations in terms of life history traits, so an increased stress tolerance, and autumnal collections would reflect more southern populations. But no one's really looked at selection on such a short time scale as a growing season. And so today I'm going to look at that using Drosophila melanogaster, and my title kind of gives away the punchline, but Drosophila melanogaster has been shown to have pronounced latitudinal declines in genes, phenotypes, and expression patterns across multiple continents. And I'm going to be comparing it to its closest relative, Drosophila simulans, to look at parallelism between species. Simulans is generally a more tropical species. As you can see here, melanogaster is found at higher latitudes and at higher frequency in colder temperatures. And when people have investigated declines in simulans, they have found far fewer declines, and the declines are much more shallow. We also see temporal dynamics in both species. So this is from a single population in Pennsylvania across 4 years. Each of these blocks is one growing season, and you can see that early in the season, the population consists entirely of melanogaster, that simulans appears throughout the summer and grows in frequency, but by the following spring, the population is again entirely melanogaster. And we're attributing this pattern to the fact that there's a well-characterized reproductive diapause in melanogaster that allows them to overwinter, but no overwintering mechanism has been identified in simulants. So it seems like there's a possibility that they are recolonizing annually from a southern refuge. And so I want to look at additional demographic parameters, and I began by looking at the age structure throughout the season. Now, in order to do this, I collected flies from the wild and then reared them in lab, and I used the full lifespan of their offspring to back calculate the age distribution of the wild flies. And this is using a model developed by Muller called the deconvolution model. And so what we can see with melanogaster is that early in the season, the flies are pretty uniformly young, but throughout the season, there's increasing age heterogeneity in the population. And this is consistent with the idea that when the environment becomes more favorable in the spring, the adults that have been in dormancy emerge in a synchronous manner and begin to lay eggs. And what we're capturing here is the first cohort that's developing early in the spring. And so based on the slower development time with the cooler temperatures in the spring, we estimate that these flies that were collected at the beginning of June were probably, the eggs were probably laid in early to mid-April. When we compare this with simulans, which was not even present in the first collection, we see that the population is already, contains a lot more age heterogeneity and is maintained throughout the season. And then I wanted to also look at different life history traits and how that, this is consistent with the idea that the populations are not overwintering and that they're coming from somewhere else, because there's no reason that populations would be age structured. I want to see if these patterns held for different life history traits, and because of the length of the talk, I'm going to focus on thermal tolerance, which was shown that temperature has strong gradients both throughout the season and also across spatial gradients as well. And so what we see is these are the same lines that I looked at the age structure, but I maintained them in a common laboratory environment for several generations in order to remove any maternal effects. So the phenotypic performance is just reflecting the genetic composition of the population. And what we see is the flies early in the season are more stress resistant, and that declines throughout the season. So the June flies survive a heat stress much longer, and they also recover from a cold shock much faster. When we look at simulants, we don't see any sort of directional change, and so the combination of the life history stress assays and the demography are creating this picture that melanogaster is a resident temporal population that's able to respond very rapidly to environmental changes, whereas simulants, which is recolonizing every year, does not respond very well to the changes in the environment. And given the pronounced seasonal selection in melanogaster, and the strong clinal variation as well, I wanted to see if there was an interaction among these. So I'm going to just look at melanogaster now. And we know that when we see variation along a cline, something occurs at a certain frequency in the south and another frequency in the north. There's the potential that these seasonal dynamics that we're seeing is actually what's playing a role in driving these patterns. And there are several ways that this could be done. The first of which is that the different latitudes could have different winter selection pressures. So when you collect the flies in the spring, there would be different intercepts, and there would be similar summer selection pressures, which is shown by these parallel lines. But when you collect the flies in the autumn here, you see that they have the frequency that you see along the latitudinal cline. Another option is that there are different the winter selection pressure is the same, there's a certain threshold, and so you have the same winter intercept, but the differential selection throughout the summer results in the same clinal pattern that we observe. And a final hypothesis, which was suggested by Romberg and his colleagues, is known as the seasonal phase cline, and in this hypothesis, the summer and winter selection pressures are the same, but what's driving the variation along the cline is the difference in the growing season. So the ephemeral growing season of northern latitudes has less time to select against the winter allele, and therefore it ends at a higher frequency. But you can see that all of these different hypotheses of seasonal selection would result in the same clinal variation that we observe. And so I tested this by collecting melanogaster from populations that are equidistant north and south of the temporal population in Pennsylvania that I looked at in the previous section. I collected both early and late and want to look at these reaction norms. And again, I'm just going to focus on thermal stress for this talk. But what you can see is that along the cline, there's strong seasonal changes, and that it's reflecting the pattern that you observe from the known data on latitudinal clines. So the northern populations are more stress resistant than the southern populations. We see that there are different patterns of selection, so it seems that there's differential winter selection occurring in the more northern populations, whereas the summer selection is what's driving the pattern in the more southern populations. When you look at Chilcoma, again there's strong seasonal selection, and the differential summer selection is resulting in the clinal patterns that we know, which are the northern populations are more resilient to the chill stress than the southern populations. And so these are really complex phenotypes. Obviously, they're undergoing selection at a lot of different levels, but what this is really exciting is that it shows how seasonal selection, both throughout the summer and the winter, can be driving a lot of the patterns that we know to be clinal. And so, in conclusion, we've been looking at adaptation to environmental heterogeneity, and we see that it's really important to consider seasonal dynamics. So in general, people have used latitudinal clines as a proxy for climate when they want to look at the adaptive process. But what we see here is that the seasonal dynamics, both from winter to spring and from spring to autumn, play a huge role in the evolutionary dynamics of the population. And additionally, even in a short-lived insect, such as Drosophila, age structure is pronounced to the effect that it's going to be playing an important role the age-specific fitness and performance will be playing an important role in the population dynamics in the wild. So I'd like to thank my lab, in particular my PI and Paul and Catherine, who's helped out a lot both in the lab and in the field. My collaborators, particularly Samuel Watson, who helped with the deconvolution model. My committee members provided useful feedback, and this would not have been possible without the orchards that permitted me to collect their fruit flies and our funding sources. So I think I have time for a few questions, and if you found this interesting, I'm giving a poster with a more complete story on seasonal changes in Drosophila on Monday evening. So stop by there or find me during a coffee break to talk more about it. Yes, are there any questions?
Did you say you estimated the age distribution in the field by looking at the lifespan of the offspring that were then hatched in the lab?
So I guess I can pull it up. So what I did is I looked at I brought the wildflies back into the lab and measured their survivorship, and then I compared that to the full age lifespan of their offspring, which were reared in the lab. So what you can see here is that the dashed lines are this is just the Kaplan-Meier survivorship curves. The dashed lines are the F1s that were reared in the lab, and the cell lines are the wildflies. And so you can use their survival patterns to estimate the distribution of the wildflies, the age distribution of the wildflies.
So what was the measure of heat resistance?
It was survival time to knock down at 37 degrees